So thank you for joining us today. It is now my pleasure to introduce our speaker, Graziella Piras from 908 Devices.
Thank you so much, Leah, and welcome everyone. Hello, everyone. Thank you for joining us today. My name is Graziella Piras, Bioprocess Segment Director at 908 Devices. And today I'm going to walk you through some important facts about cell culture media analysis. So why should we care about cell culture media analysis in bioprocessing? We'll talk about the amino acid impact on cell growth, productivity, and cell health. And I will start to shed more light on why we should care about cell culture media analysis. We'll see some examples of spent media analysis and improvement of productivity and protein quality. And then we'll talk a little bit about cell culture media optimization strategies and typical upstream process analytics. And you may wonder, well, how easy is it to measure key nutrients in my medium? And I will answer that question. We will then finish with a high throughput spent medium analysis case study for map process understanding. So in the upstream process development work, there are three major components that will help you achieve those high titers, low cost of goods, and desired CQAs, protein quality attribute, to generate safe and efficient drugs. Of course, the cell line is very important, but also the bioprocess parameters and the bioreactor process and parameters, and then the cell culture medium, which is at the center. And we know, especially with chemically defined medium, it's very important to know what's in the medium and being able to adjust those components. So let's dive into it. So we know that amino acids are central nutrients in cell, mammalian cell culture. They play many roles. Of course, they are the protein building blocks. They also play a major role in cellular metabolism. On the far right here, we see that, for example, certain amino acids are used as an energy source like glutamine and enter the Krebs cycle, TCA cycle. Amino acids also play in the biosynthesis of themselves, of nucleotides and other amines, and are involved in many other metabolism. And they can also play a role in inhibiting or enhancing some enzymes, and they play also a role in the transport of themselves into the cells. So lots of very key roles in the cells. So if we think about amino acids and cell culture medium now, growth, titer, and protein quality attributes. Amino acids can provide a protective effect against ammonia, osmolality, high osmolality, apoptosis, or dissolve carbon dioxide. In amino acid content can alter cell growth, viability, and productivity. And finally, amino acid can impact protein quality attribute. So let's look at some examples. So does it really matter to really look at, the details of amino acid composition and consumption? Yes, it does. Here is the first example from a very nice paper published about 6 years ago, where it was demonstrated that the balance of glucose and amino acid is critical for cell growth, IgG titer, and also glycosylation patterns. And we can see here that different types of media and feeds on two different cell lines gave widely different titers. The authors also showed that some undesired high mannose 5 glycans were linked with depletion of glutamine and extracellular environment. In the next example, we have control of misincorporation of serine for asparagine using spent medium analysis. And we see in this paper that when asparagine is depleted from the medium, we have misincorporation of serine. And the authors did a very nice work in restoring protein quality by balancing out these two amino acids and some other ones as well. So another example here of the impact of amino acids specifically on protein quality attributes. In the third example, another very nice paper published about 6 years ago as well, where the authors develop a strategy for optimizing defined feed using spent medium analysis to improve MAP titers and reduce lactate. Of course, we all want to improve those titers, and we know that lactate can be detrimental to your culture, and we always want to keep that lactate level low. In this example, the first graph on the left, spent medium analysis was used to identify specific amino acid depletion. So we see asparagine, glutamine, serine, cysteine, and tyrosine being depleted during the time course. What the author did was testing several types of supplementation with these 3 different amino acids in different combinations, and they found that supplementing all 3 amino acids together gave them the highest relative increase in titer, and also gave them the best control of the lactate, so a higher lactate consumption toward the second phase of the culture. And they obtained about a 60% increase in titer. So we can see through these examples that monitoring amino acid composition and feeding is very important for many aspects of the cell culture. So you may wonder, well, what is the role of each amino acid? And we know essential amino acids are key. You have to have them all, but also non-essential amino acids need to be well supplemented into your culture. So they provide protective effect, as I mentioned before. And there are many examples of specific amino acids. For example, we know that threonine protects the cells against cell stresses and against a high level of ammonia, for example, just to give you an example. For cell growth, well, all essential amino acids should be at high levels. Tryptophan, for example, needs to be always, at good levels, but it degrades in the presence of fluorescent light. So it's one that you need to really watch out for. Non-essential amino acids are also important. Tyrosine depletion, it's not an essential amino acid, but its depletion can hurt specific productivity, for example. Another specific cell line that requires specific amino acids, like glycine for CHO-DG44. And then amino acids specificities for protein quality attributes. I showed the example of the serine misincorporation at asparagine sites. Tyrosine and phenylalanine misincorporation can also happen, and so these need to be well balanced as well. And then lysine and histidine can have an impact on charge variant. And these are just some examples. There's lots of examples in the literature. Actually, I listed here a review that talks about all these different details. So how can you optimize the amino acid for your cell line? So a typical process would be to first ensure that you have medium consistency and do a medium QC. So if you are getting your medium from a media vendor, for example, or ordering your custom media, making sure every lot is consistent, is key. And sometimes you may find that it's not always. So checking amino acid and vitamins in your cell culture medium is one way to do that. You want to screen, if you're developing a new process, you want to screen many media and feeds with diverse composition. Cell line specific commercial media offer a quick way to screen multiple media. And you may want to make sure that you're testing media that offer a diverse set of nutrients. You want to perform spent medium analysis during your fed batch time course so you can identify the key components that get depleted or that are overabundant, which can also happen and is not a good thing either. And then you want to remedy depletion or overaccumulation by developing your feed strategy, supplementing feeds or your media, and maybe go into a DOE to maximize your experimentation. And then finally, once you have developed your media and your process, you may want to use also spent medium analysis to monitor cell health and make sure that everything is going according to plan. So there are many ways to use, where cell culture media analysis matters, specifically amino acids and vitamins, which are key. I've listed here process intensification with perfusion processes. Definitely, the medium is central. You want to find the balance between nutrients and your process. Even in fermentation, amino acids, supplementation of amino acids can lead to less cell stress, and better productivity. In CAR-T, I'm not going to talk about it but at length here, but it's known that specific amino acids are required for cell health and efficacy of the CAR-T, and there are many examples throughout the literature. So how do you measure your cellular and extracellular environment today? Of course, for cell growth and health, you have your great instrumentations to measure viability, viable cell density, cell size, key metabolites. Those are very well established and are at line, sometimes online as well, and are very quick measurements. Your parameters usually are also online. The amino acid and vitamin, although they're key, you know that usually you have to take samples and send those to a core lab or a contract lab to get these measured, and it may take several days, and I know sometimes it even takes weeks to get your data back. So it's a little bit counterproductive since these are so important. So a solution that 908 Devices has developed to help here is we have developed an analyzer, the Rebel, which provides basically measurement of amino acid and water-soluble vitamins at the point of need. Very small samples, a broad range of analytes. You can measure in less than 10 minutes, and the Rebel is very easy to operate. All you need to do is being able to pipette, basically. And here are the analytes, so all the amino acids, some water-soluble vitamins, and also some bioamines. So in terms of strategies, if you're in early process development, you may be looking at using a commercial media and feed. Remember to QC your media, and you may want to screen a variety of media. And if you use the Rebel, it'll give you sort of a landscape of amino acid and vitamin composition, so you can ensure that you're testing various media. If you're using a platform media, maybe your company has a platform media. Again, you want to maybe do some limited optimization early on, and so the Rebel is very useful. In late process development, where you're going to full optimization, definitely, again, spend medium analysis leading to DOEs for a full optimization and really providing those key nutrients to your cells when they need it. So I want to show you briefly a study that was published, an initial study published in last year, and then a follow-up that was published recently in 2021. This group here, this is a collaboration between Clemson, Heidelberg University, and Tulane University, and they used the Rebel in tandem with the Amber in tandem with the Rebel, and they were able to generate very nice data to show how cells can detoxify from high ammonia, which is usually produced towards the end of a culture. It can be have negative effects on your protein quality attribute, and also on your culture health. And basically, they were able to measure all the amino acids throughout their cultures in all their different samples, and being able to show that alanine specifically was overproduced in the high ammonia cultures. And they were able to demonstrate or show that alanine production was the primary mechanism that the cells used to detoxify. So finally, in summary, I would like to say again that, measuring what matters when it matters is key. And the Rebel is a great instrument to have in tandem with your bioreactors, your microbioreactors, to ensure media conformity, bioprocess monitoring, viral vector production. Didn't have to talk about that, but of course, that is, viral vectors are also, produced in cell culture, and amino acid and vitamin optimization in central, and then briefly touch on CAR-T. So I would like to thank you all. I would like to thank Bioprocess International for hosting us, and I would be happy to answer any questions. Thank you so much.
Okay, great. So the first question is, are commercially available medium and feed not already optimized for CHO or other cell lines?
Great question. Yes, the commercial media that you can find from many different vendors have been optimized. That is correct, but they've been optimized on the cell line that vendor, is using. And so to further refine, maybe that your cell line has a specific key nutrient that needs to be optimized, or you're looking at a specific quality attribute that needs modulation or specific balancing of some key amino acids. So I still recommend, even if you're using a commercial media, to do spend medium analysis. And also for your feeding strategy, it's very important to be able to know when is best to feed and how much.
Okay. Is the Rebel connected to a microbioreactor such as the Amber 15?
At this moment, it's not connected. It can sit right next to it, and you can basically pull samples aseptically and load them on the Rebel very easy. But at the moment, it is not. But it is, of course, something that would be of high interest.
And is there data analysis software for the Rebel?
Right, great question. Of course, you may generate a lot of data when using the Rebel. And so the Rebel will produce a very nice report on all the amino acid and vitamin analysis that it will perform for you. And that data in the form of a CSV file can be exported into any Excel or any software. But we have actually a special connection with the Simca software from Sartorius, where you can do your multivariate analysis.
And is there a benefit in performing cell media analysis for a perfusion process?
Yes, there is. If you think about, the cell culture media in a perfusion process where you may be perfusing, 1 or 2 vessel volumes per day, and you want to keep that CSPR, so that cell-specific consumption of media as low as possible, definitely we see a lot of work being done to balance those key nutrients so that you're not wasting medium, but you're also giving your cells the right, nutrients so that you can sustain those very high cell densities. So definitely in perfusion is very important to also monitor your key nutrients. Absolutely.
And how much expertise do I need in analytics to run the Rebel?
If you can pipette, you can run the Rebel and dilute your sample. It's basically designed to, make your life as easy as possible so that you can get the results you need when you need them. So it's very easy to run.
Okay. And how often should you measure AA and vitamin during a time course?
I would recommend at least daily. With the Rebel, you could do several times a day, which is almost nearly impossible with the traditional method because of sample volumes, but also, it's costly. You have to send your samples out. With the Rebel, definitely on a daily basis, so you can see the progression and you can see the nutrient balance with your feeding. So definitely on a daily basis.
Okay. Can I measure AA viral vector process with the Rebel?
Definitely. I didn't have time to go into it, but one of the key challenges currently in viral vector production are to increase those titers in those transient transfections or even stable, cell lines. So it is very important to balance those amino acids to achieve those high titers.
Do proteins need to be removed from samples before analysis on the Rebel?
Very good question. So actually, no, they don't. All you need to do is spin your samples, remove the cells, or filter your sample. And no need to remove any proteins.
Okay, so it looks like we're running out of time for questions. If we didn't get to your question, we will be passing those along to Graziella and she can follow up with you directly. The last question for this webcast is, I'm developing a transient process to produce AAV. Any suggestions you can provide to increase viral vector titers?
Right. So with the transient transfections, there are many components that come into play. You have your amount of DNA, your ratios of DNA, your ratio of PEI and DNA. And we find that also the type of media you're using plays a significant role. So I would advise to screen, or if you're starting a new process, as you probably know, there are a lot of commercial media available for HEC293 cells. A lot of times those media have been developed for protein production, but you can start there and then see how you can improve those media based on your process.
Okay, well, thank you, Graziella.
Thank you so much, Leah, and thank you so much, Bioprocess International, for hosting us.
And thank you to our audience